This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 571, recorded on October 25th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in a rather cloudy New York City, Dickson Despommier.
Hello, Vincent.
17 Celsius.
Yep. It's not bad out.
It's fine.
It's not good out. It's just like, meh. It's a meh day.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here it's also cloudy. It's 48 Fahrenheit. And my quick app to convert to Celsius, okay, that's
not very quick.
Almost 9 Celsius.
Ah, it's chilly.
Yeah. Chilly.
From Madison, New Jersey, Brianne Barker.
Hi, everybody. It's great to be back. It is also somewhat meh here, but maybe less so. I would guess it's only partly cloudy. Yeah. And 18 Fahrenheit.
Well, we got the heat bubble.
Right.
And from Western Massachusetts, Alan Dove.
Yeah, good to be here.
Is meh, is that Hebrew? Yiddish?
No, it's just current.
Yeah.
It's current?
It's like a New Yorker.
Well, in fact, it's several years old.
It's
yeah.
So it's new, yeah? Really?
It could be.
Let's look
it
it up.
It is frequently used to mean mediocre, particularly today in my class when I was talking about the strength of certain types of immune responses.
Well, that's good.
They were meh.
A meh
They
response.
were. Yeah.
Yeah, the origin, some have speculated its origin is Yiddish because of similarity to feh.
Yes.
Which appears in the 1936 Yiddish song.
So, Brianne, all those weak immune responses are generated by macrophages.
Exactly.
Macrophages.
Macrophages.
Okay, well.
And it's pretty much the same weather here. It's overcast, 17C.
If you like what we do here, if you don't think we're meh.
If you want us to stop doing the weather, you'll have to support us double.
Consider supporting us financially, microbe.tv/contribute. This first follow-up, Dickson, I think you should.
I guess.
Even though it's very long, I know it will tire you.
I'll do it. Follow-up. Chris writes, hello, Twivers. It was a beautiful, sunny and windy 60F here today in Stony Brook, Long Island. I typically only write in for TWIP. I'm the one who likes to send pictures and videos of fish parasites. But I am emailing you because I am the one who corrected Dickson on his not wrong, but not right information concerning iron in the ocean. To clarify, Dickson is right that iron is limiting to phytoplankton growth in many areas of the ocean. But the point I made last time was that iron is only limiting in certain areas of the ocean, specifically the open ocean, which includes about 99.9%, but that's okay. So areas such as the coast, and that's changing, of course, with climate change.
Hey, Waldo, doesn't apply.
That's right. Exactly. So areas such as the coast are not typically limited by iron due to the close proximity to land, which is a major source of iron. As you move away from the coast, iron will become more limiting, but these areas can also be limited by other nutrients such as nitrogen and phosphorus. Areas that are most depleted in iron are the high nutrient, low chlorophyll, HNLC zones, such as the Southern Ocean. Then these areas have large amounts of upwellings, bringing up nutrients, nitrogen, but are relatively removed from the coast, so they are limited by iron. These HNLC zones are actually where the iron fertilization experiments took place. So I didn't say Dickson was crazy. He spelled my name correctly, and then he misspells my name. But I'm not being critical, mind you.
That's because I corrected most of them, but I missed one.
Oops.
Yeah.
Oh, good. Okay. But that he was slightly incorrect in saying that all the ocean is limited by iron. Furthermore, Dickson mentioned harmful algae blooms, HABs, that are limited by iron, but because HABs are defined by the damage they do. Specifically, they damage in relation to anthropogenic resources, fisheries, water resources, etc. They are typically on the coasts, so HABs are rarely limited by iron, but excess nutrients, primarily nitrogen from fertilizer and wastewater runoff, typically cause these blooms. Sorry to be nitpicking, but as HIBs become even more prevalent with climate change and such, it's important to clarify these details. Additionally, I would like to add to Dickson's comments about William Campbell with a short story. I met William Campbell at the Helminthological Society of Washington meeting directly after he had received his Nobel Prize. He was arguably the most delightful scientist I have ever yet to meet. At a small meeting, less than 100 people, he made time to talk to every undergraduate, which I was at the time, even though he had just won the most prestigious award a scientist could. Furthermore, upon talking to a fellow lab mate who was interested in veterinary parasitology, he offered to send one of his own personal textbooks from his library to a sophomore he had just met because it could be a valuable resource for her. Truly a nice man who has done wonders for this world, and I look forward to hearing the TWiP Heroes section that I'm sure you will write about him when we've done that already. Thank you. Always loved the episode, though it was one of the most interesting I've heard yet. Warm regards, Chris. Yesterday.
No, that's mine.
Oh, sorry.
Sorry. I should have made a little section divider there.
Okay.
I thought you were one of the nicest, most delightful scientists in the world, Dickson.
I would love to think that, but I don't think it's true.
Well, actually, you haven't met Chris. He might think you are.
I would love to meet him. I would.
Yeah. Well, we may go out there
He's not too far away.
To record. Apparently, there's some people out there who would be interesting.
Oh.
On all of Long Island, there must be some.
Must be somebody interesting.
Oh, no, come on.
No,
Cold Spring Harbor's out there. Come on.
Just kidding.
There's a bunch of stuff out there.
So yesterday was World Polio Day. And the WHO took the opportunity to announce the eradication of wild poliovirus type 3. And if you go to the site, there's a little certificate. Your virus gets a certificate of eradication. You go to polioeradication.org, and it's certificate of eradication signed by 6 people of all the regions, the European, African, Americas, Eastern Mediterranean, Southeast Asia, and Western Pacific. It's pretty. I just think it's funny that there's a certificate. Like, the virus will never see it because it's eradicated.
It's gone. That's right.
Yeah,
anyway,
does the virus have eyes?
Yeah, right. So this is interesting. So wild poliovirus, because the vaccines are still being used that are infectious, so you can't say all polioviruses. So now it's wild types 2 and 3, which leaves only wild poliovirus type 1. And this, of course, is as far as we know.
And, of course, it leaves the vaccine-derived strains.
Yeah.
All right.
And as far as type 2 vaccine-derived strains, they're going crazy. There are just 100 cases so far this year of paralysis caused by circulating vaccine-derived type 2. Yeah, it's crazy. But we'll see what happens. Anyway, that's cool. We have a snippet which was inspired by Frank, who writes, Dear League of Extraordinary Virologists. It's hard to believe that the fascinating podcast I started listening to in 2008 is now on episode 568, and we all seem to be just as young as sassy as back then. It must be due to better life through virology. It's a good bumper sticker, right?
Yeah.
That is a good bumper sticker.
I remember being impressed with your early description of recombinant flu vaccines as having the ability to produce a protein which could not be accomplished through egg production. The ability to produce vaccine in a shorter time, closer to flu season, also had the obvious potential to address the most recent strains. I believe this discussion was prior to clinical trials. Can you give us a timely update on the advantages of FluBlock, which has become commercially available, breadth and level of protection, age appropriateness, etc.? Thanks for keeping us young and healthy, Frank in Sausalito.
Just
but
north of San Francisco.
Shouldn't have an Italian accent, I know. Sausalito. Sausalito. Yeah, just north of lovely little town, right?
Great town.
Mm-hmm.
Absolutely great town. Mm-hmm. Yeah.
So I thought we'd chat a bit about FluBlock. There's a CDC page. If you'd like to explore it, we'll put a link to that. FluBlock is hemagglutinin protein produced in insect cells using baculovirus vectors. And this just brings to mind what Kathy said recently. The person behind you is the best baculovirus virologist in the world. It's so great.
Yep.
So you make the HA proteins. It's quadrivalent, so 4 different HA proteins. And you purify the protein, and that's the vaccine. And it has 3 times more HA protein content than the other vaccines that are.
Give us the short version of why insect cells were used. Because baculovirus works best in insect cells.
Well, it's an insect virus, right? Optimized for.
And baculovirus produces huge amounts of protein.
And you don't need animal serum.
Right.
So it's cheaper. Dickson, you asked the question. Look at me. It's cheaper. No animal serum.
Cheaper.
It's much easier to do huge quantities.
Easier to purify.
Well, if you don't have all those serum proteins, I guess it would be. But it's cheaper also. So there are many reasons. Many proteins.
Also, it's pretty safe since it won't infect humans.
Yes. That's right. It's just HA protein.
Yeah.
All right. So let me tell you a few things about this.
And you can do it all at room temperature.
You can.
Yeah, that's right. That's right. I've seen people's labs who work on baculoviruses. They're just sitting there, you know.
And I don't think they use CO2 either in the incubators.
Don't need CO2.
Look at that.
Yeah, insects are just advantageous.
Probably the medium is cheaper because you don't need bovine serum albumin.
And as Frank said, it's not grown in eggs, so you don't need to make egg-adapted strains. You can respond quickly if there's a brand new. All you need is a sequence of the HA. So there are lots of advantages. But what about its effectiveness? So I have a paper here in the Journal of Infectious Diseases, which is called Randomized Comparison of Immunogenicity and Safety of Quadrivalent Recombinant versus Inactivated Influenza Vaccine in Healthy Adults 18 to 49 years of age. Now, this is immunogenicity and safety. It is not efficacy. However, the trivalent preparation of Flublok, which was the first licensed, it's made by a company called Protein Sciences, which is in Connecticut. They did an efficacy trial of that some time ago, and they compared it to licensed inactivated vaccines. It was tested in both 18 to 49 years of age and older age group, like you and me, Dickson, the older age group, although you're much older than I am. And in those studies, they found that the efficacy supported non-inferiority and superiority of the Flublok versus. I don't understand that. It's either one or the other.
Do they ever say superiority? Because the very first time I ran into endpoints for non-inferiority, I underlined it and put a question mark by it.
Right.
And when you put them two together, you get a meh.
Yes.
Well,
non-inferiority is meh, I think.
Well, it's not worse. It's not worse.
It's
not
just
worse.
not worse.
It's either as good as or better, which is what you would need for licensure, right? And so I don't quite understand the numbers. So they say, well, in the study with older people, the relative vaccine efficacy versus the inactivated was 30%. I don't know what that means, right? Because on its own, 30% would be horrible. I don't know what 30% means, but it says it satisfied this non-inferiority criteria. And the other one in the other age group was 43%. Anyway, on that basis, this was licensed, the trivalent, okay? So all we know, and there's been nothing since I looked, there's been no other efficacy trials. The quadrivalent, you have flu?
You don't sneeze with flu. Even I know that.
So the quadrivalent, this paper actually has a study of the quadrivalent immunogenicity and safety because it's a new formulation. In fact, now all flu vaccines are quadrivalent, no more trivalent.
Right.
Quadrivalent. If you sneeze twice, you'll have a quadra sneeze.
So the quadrivalent
for the trifecta.
gives you the 2 current strains of influenza A and 2 strains of influenza B.
Right.
So
basically, the only available efficacy of the Flublok versus traditional inactivated said it was comparable.
And that was trivalent.
The trivalent. Now, for this quadrivalent, they say it is safe and the immunogenicity is also comparable. Right. So the conclusion is the first head-to-head comparison of recombinant versus inactivated quadrivalent, 18 to 49-year-old adults showed comparable immunogenicity, safety, and tolerability of both vaccines. So we don't have an efficacy trial. So I'm not sure we will actually have one.
I'm not sure that saying something is... Oh. I was going to say, if something is non-inferior and tolerable, those aren't really,
It's not a great ad.
remarks.
It's not a wonderful sale.
Exactly right.
But you got to start somewhere. And these trials, the efficacy trials, are really tough to set up.
Of course they are.
Well, one of the problems is that now we understand that every person has a different history of flu vaccination and infection, and that influences how you respond. So I don't even know how you would do a proper.
There's that. There's variation from season to season. There's what are your endpoints? What do you mean by efficacy? And we've talked about all these things a lot in every time flu vaccines come up. And then if you're going to compare two vaccines, you've got to go through all that double.
Yeah.
So that's it takes a while to get around. I'm sure at some point somebody will do the comparative efficacy study on these. But the priority was, is this thing safe and does it have the same immunogenicity? And the answer is yes.
So I would, if you have a choice, I think this is fine, right?
Sure.
It has more antigen in it. That's fine. NIH is currently carrying out a phase 1 where they're going to compare this vaccine with and without adjuvants. Early stage evaluating 2 licensed vaccines with or without adjuvants.
What's the adjuvant?
2 different. One is an aluminum salt, and the other one is I think it's a carbohydrate. Sugar-based.
Yeah. Although it says CPG in the name.
Look at that.
So
yeah.
It makes me think that it has some kind of
DNA.
Nucleic acid.
Yeah.
Mm-hmm.
Yep. Well, 2 bases anyway.
And I'm guessing that stems from the discovery back when antisense therapies were all the rage, and there was this huge bubble of investment in everything's going to be cured by antisense. And then people discovered that the efficacy they'd seen in the trials with antisense was really due to the CPG response.
Exactly. The DNA sensing. CPG as a PAMP, as a spoiler for the other paper.
Yes.
So the way this trial is going to be. So
CPG, I'm guessing this adjuvant is taking advantage of that and adding CPG as a pathogen-associated molecular pattern that the innate immune system then reacts to and fires everything up.
Exactly.
So they're going to compare Fluzone and Flublok. So Flublok is the baculovirus. One Fluzone is probably a traditional one.
It's a standard, yeah.
And Flucelvax is propagated in cells and culture. And then in
Fluzone is Sanofi's. That's probably the most produced flu vaccine.
Flublok, Fluzone. Yeah, so then we'll do 2 different ones with and without adjuvant. So that may give you an indication of the efficacy of Flublok, right?
Yeah.
With and without adjuvant. So that is just enrolling phase 1. So it'll be a few years. And Tony Fauci is quoted as saying, we like to make new flu vaccines, but also improve the ones we already have. To which someone would say, well, I'm not taking it till you improve it.
Right.
It's a fine line, isn't it?
Very.
I mean, I'll take it anyway, but I can see people saying, hey, if you're not done with it, just call me when you're ready.
Yeah, but
I'll take it anytime.
Yeah, are you going to postpone buying a computer until they're perfect?
Exactly.
You're just being logical, Alan. Hey,
I know. It's an emotion thing that people are afraid of needles. Yeah.
Dixon and I will go next week to get our.
Are going to do the.
So
right.
I'm a little confused by this NIH study article. It says they're evaluating 2 licensed seasonal influenza vaccines. So that means this quadrivalent one, the Flublok, is already licensed, but they don't have efficacy data.
They have efficacy data for the trivalent, but not the...
Well, they have is immunogenicity and safety and tolerability. That's right. No efficacy data.
So they can license it without the efficacy data.
Apparently.
Huh. Okay.
I think because it's shown to be non-inferior, they can.
Well, and I think because the efficacy was shown for the trivalent.
Yeah. I mean, I'm just a little bit surprised.
It is. Yeah, that is a good question.
The other paper I found, which is interesting, is by... is in the Journal of Virology. It just came out. Monoclonal antibody responses after recombinant hemagglutinin vaccine versus subunit inactivated vaccine, a comparative study. Now, this is not efficacy, but they compared the 2 vaccines that we're talking about. They look at the antibodies that are induced in people. So they immunize people. They take B cells from them and clone out the monoclonals and produce the proteins, a lot of work. And then they see how the two vaccines do, and they're pretty much similar. They both do mostly head domains of the HA antibodies rather than stalk, which we know are the more broadly neutralizing. And they do the same experiment in mice, which have no previous influenza history compared with people, as far as we know, right? Mm-hmm. And you can see the same, they get similar results. So at least at this level, in terms of inducing monoclonal antibodies, the two are very comparable, which means the HA is similar, right? Mm-hmm.
Yeah, the flu cell vax, at one point they call it a subunit vaccine, but then somewhere else they say virions produced in mammalian cells.
Yeah, I think they break it. They when they inactivate it, it is made in MDCK cells, right? There's virions, and then they break it up.
Okay. So
that's why they call it subunit. But I'm not sure they do any purification, but they're just pieces in there. So
okay. Somehow I missed that fact too. Somehow I'm really annoyed with papers lately where I just don't think they're explaining things well. So yeah. Okay.
All right. So that's that. And then a couple of other things I wanted to point out. So there's another page at CDC with the current recommendations, which is very helpful if you want to see what you're getting this year, which is all quadrivalent, et cetera. And the attenuated vaccine is now available again, the nasal spray, which had been absent for a while.
Right.
And then the other day, my wife calls me. She says, I'm at the pharmacy. I'm getting a flu vaccine, and I'm supposed to get the high dose, but they don't have any more.
Right. So get two small doses.
Get the low dose. And
that's what I would do.
I said, I'm not sure it makes a difference anyway. So I looked to see if there are any efficacy data, and there aren't any for high dose. So 65 and over, you're supposed to get the high dose, right?
Yeah, but they don't list a good reason as to why.
And the only paper I found was a paper in 2019, just this month, immunogenicity and safety of high-dose quadrivalent flu vaccine in Japanese adults 65 and older. And they say the results add to the evidence that high-dose vaccines are more immunogenic than standard dose in this age group, but they don't do efficacy, right?
Nope.
Although I guess that's part of the issue in being immunogenic, right?
Yeah, but wouldn't they be more immunogenic in any age group?
Because you're giving more of it?
You'd expect so.
Mm-hmm.
In fact, I kind of thought that was the whole point.
No pun intended.
I mean, yeah. So you know, 65 and older, you need to give more antigen to get a similar immune response, right? That's the idea. But yeah, if you gave it to a younger person, it wouldn't hurt, right? But it's kind of a waste of antigen, I suppose. And obviously they run out of it, so.
Antigen is wasted on the young.
That's good. Another good bumper sticker.
That is another nice one.
Antigen is wasted on the young. Dear. So this year, Dickson, you and I will get high dose.
We will.
Okay. That's fine. I just don't know if it makes a difference, right? There's no data. So someone just decided putting more antigen.
It just seemed like a good idea.
Let me look at this paper again. What did they actually find here?
I think the data are based on the immunogenicity. There are data. They're just maybe not efficacy.
So these here, adults over 65 were randomized to get
regular or high.
Standard or the high dose. Okay. Irrespective of the administration route. So they tried sub-Q versus IM. Post-vaccination, HA titers and seroconversion rates were higher for the double dose than the single dose. Okay,
right.
So you get higher titers and higher. That's good. So
Doris could have had an injection on each arm and gotten the higher dose. Double.
Yeah, I mean,
or gone to two different drugstores.
Exactly.
Right. Get a second shot.
Yeah, it's true. But I told her, don't worry about it.
There was a second shooter.
I told her I wasn't convinced of there's no efficacy data, so, and there aren't.
No, but usually there isn't anyway, so.
It's for the trivalent flu block.
How good is the epidemiological data that shows that when you did have the right strain and you did immunize, that the flu outbreak was lower?
Yeah, they have good data. They collect it. It's all on the CDC webpage. You can see they have vaccination efficacy every season for sure.
Right.
They do studies.
And it's randomized.
Where they look at people who are vaccinated or not. Yes, it's all there. You could actually go now and look, but I don't want to take the time to do it. Maybe next week.
What about the mortality rate of people over the age of 60, which is like 40,000 people a year die from?
Well, what they have is they have a thing called excess mortality from pneumonia. So usually that's what's killing you with flu if you're older, right? So they track that, and you can see in some years how it's higher than in other years, and that's a particularly nasty flu year. Yeah, that's what they track, because when you die of influenza, that's not usually what they put on your death certificate. They put pneumonia. That's right. Something else. I only saw one death certificate, and it had death from this, secondary to this, secondary to this, secondary to this. That was so weird.
Wow.
Yeah. Okay. Enough flu? We have actually a letter, but we'll...
Yes, enough flu. That's what I can say right now.
Do you have the flu?
I am not sure what I have, but whatever it is, it's unpleasant.
Brianne, you're a virologist. You can find out.
I could.
Just take samples. Grow them up.
Yes.
I speaking of diagnostics of flu, which is what you would take a nasal swab, right?
Yes.
I follow a young lady on Instagram who's a laboratorian, and she goes around and gives talks to practitioners and nurses on how to do a proper nasal swab, because apparently it's not easy and people screw it up. You can imagine they don't go in far enough; they go too far, right? So it's an art like anything else. All right, we have a paper which came via Ed Grow. So Kathy actually forwarded his tweet, because I've had my notifications turned off for a while. He wrote, koalas are one of the only models for real-time invasion with endogenous retrovirus endogenization. Check out this fantastic work on piRNAs in action, a good paper for TWiV. And then later, Chaim sent, it strikes me as early work that's going to need a lot of follow-up and validation, but still pretty cool. So there you go. We're doing it.
Yeah.
The PI.
Yes. Before we dig in, I wanted to make a comment about the accessibility of this paper. It is behind a paywall, but it wasn't for a while.
Yeah.
What happened?
Okay. Did other people have this experience? Because I last night I went to the the notes for this show. I said, "Oh, okay, paper," and then I clicked on the link and I saw sell. I said, "Oh, great! I'm going to have to get a copy of this from Vincent, but I'll check the full text link anyway." And there was the full text. So I went ahead and downloaded the PDF onto my iPad and said, "Okay, well, I'll get to that tomorrow." And then today I went to download it on my desktop, because I usually for TWiV papers, I prefer to have them on the desktop at the same time, and hit the paywall.
Interesting. Maybe they made a mistake.
Yeah.
Something made me think that it was open access, and I wrote that at the top. And then on the printed copy that I have, dinosaur that I am, and then because I printed it black and white, I was in the library, so I tried to pull it up on my phone, and I was going to have to log in and So they changed it.
I have the explanation.
I think they had the door open for a while, and then they decided
Alan,
it shouldn't.
The explanation is that they wanted to rope you in with a soft sell, but now they've got the hard sell.
Well, yeah, either way, the wind blew the door shut, right?
It did.
Yes, the wind blew the door shut.
Influenza.
I know there are some cases where things are open access for the first 90 days or so after publication, so maybe we hit right at that time point.
Well, it just came out.
October 17th, 2019.
Yeah, so this just came out. Anyway, the piRNA response to retroviral invasion of the koala genome. The first two co-authors, Shanzhong Yu and Birgit Kopech, and then the last author is William Thurkoff, and they are coming from Tongji University, UMass, University of Massachusetts Medical School in Worcester. And
which I assume is Jeremy Lubin's.
That's right.
Credit.
Right.
Yeah, that's right. And the University of Queensland, Brisbane. which I assume is the source of the koalas, or
Yes,
at least
indeed.
part koala parts, let's say. So piRNAs, we have not really talked about Piwi interacting RNAs. Piwi, PIWI, piRNAs for short. These are part of the small RNA repertoire of the cell. We've talked about microRNAs that can regulate gene expression, and in some organisms can even be antiviral. These are short RNAs that combine with a Piwi protein that's kind of like an Argonaut that then degrades the target. These are thought to be around to guard against transposons.
That's right. Exactly.
Transposons, mobile genetic elements.
Fantastic.
That can move around the gene. First of all, they can come in. If it's a retrovirus, that's a kind of transposon. Once its DNA can get in, and then they move around.
You know what I think of when I hear this, Vincent?
What?
I think of the movie The Matrix. I really do. And I see these monster machines trying to invade the inner...
With those long things behind them, right?
That's
right. That's
exactly.
true. That's true.
Exactly.
Well, they're invading us.
I think we talked about Piwi RNAs once before.
Yeah.
On episode 482, because that was the first time I'd heard of them.
Well, okay.
So that's how I learned about them, and now I've gotten to learn much more.
Piwi RNA. So the idea being that, you don't want transposons jumping around the genome, right? Because they can...
That's their job.
And I don't know about their job, but they're parasites, and they just do that.
They're parasites.
And they can disrupt genes and cause all kinds of problems. So, and particularly in a germline cell, right, that would be really bad. So Piwis are thought to be really important in the germline. So and there's lots of evidence that these are important for guarding against transposons. But what we don't know is how, when a transposon invades initially, how the piRNA pathway is acting. And that's where the koalas come in, because they're being invaded as we speak, right? They are being infected by a gamma retrovirus, the koala retrovirus A. There's A and B.
And do we know where this came from?
The idea is that it came from a rodent. There's phylogenetic evidence that it's a rodent virus that jumped into koalas.
And do we know how long ago?
So there are museum pelts from the 1800s, koala pelts, and you can find viral DNA in those. So at least 100 so years, but maybe longer. We just don't know. So these
Australia's got a strange wildlife situation.
They do.
Were there rodents there before humans?
You mean marsupial rodents?
Well, would they be rodents?
They would have to be marsupials.
They would have to be marsupials, but then...
Are there marsupial rodents?
Are there marsupial rodents?
I spent 6 months in Australia and I wouldn't know the answer.
I know that the rodent source of this is in Australia, so...
Right, but everything's now. I mean, they've had been invaded by so many things. So I'm just wondering if this is... Because what we're going to talk about is that this is an ongoing invasion of this retrovirus into the koala population that has spread from north to south.
Yeah.
And there are koala populations in the south of Australia that don't have this virus.
Right.
They haven't been exposed.
So all the spread among koalas is either horizontal, right, from one to another, or vertical, the germline. So it's spreading throughout. It started in the north of Australia, and of course that's where koalas are from originally, right? So they're from the north to the south, and only a few at the south, and there's some islands off the south with koalas, so they're not yet infected, but, if they can contact mainland koalas, they'll get infected at some point.
And this is not a harmless virus.
It's not a harmless virus, no. It's immunosuppressive, so these poor koalas get leukemia. They also get opportunistic infections.
Chlamydia is their biggest problem, as far as
I talked with a guy when I was in Australia. I did a podcast with a guy who's trying to immunize the koalas against the chlamydial infection, where they take these long poles with syringes on the end, and they kind of
they won't
shove them up into the tree.
That's right.
I don't know how effective that is.
That's true. Well,
but so this is a problem for the koalas. It could wipe them out. Yeah. Yes.
Evidently, there are marsupial rats and mice but
Okay.
it appears to me that they're not in the order of rodents.
No.
So
No,
Right.
they're marsupialis or something like that.
right.
So these animals, which are in recent years being infected or invaded by these transposons, it's a good system to study what's the situation with the piRNAs, right? And so that's what this paper does. It's basically a sequencing paper where they take either database sequences from koalas that have already been established, or they do their own sequencing of RNAs, both long and short RNAs, right? So mRNA versus short RNAs, which would include piRNAs, and they compare them. They do a lot of analysis. It's a computational biology paper, and it's quite remarkable that they can actually probe the data so much. So they have they say, how many, so when a retrovirus infects, the RNA is converted to DNA, which then integrates, right? So they say, what's the status of the koala genome with respect to these integrations? And they have they have a reference genome of a koala, which was established some time ago, right, by other people. And they can see in that genome 7 full-length copies of this koala retrovirus A, core VA, let's say. Very similar, not much sequence divergence, which means it's probably been moving around recently. If it were a long time ago, you would see much more divergence among the different copies that are around the genome. They also found 3 endogenous retroviruses of different sorts, ERVs, and they give them different names.
But they're all on the same chromosomes also?
Probably not. But
No, because that's how one of the reasons they say that they are relatively recent is that they're in different locations in different koalas.
Okay.
Yeah. So they do some RNA sequencing on tissues, and they can see that the RNAs are expressed in multiple tissues. So they're active at this moment. They're not silent. They're being not only present, but they're being transcribed into RNA. So, if these things are moving around the germline, what's the matter, Dickson? You hear some hissing? That's Brianne. Yeah.
What? I'm hissing?
Yeah, there's always, and Drew, there's a background hissing.
Oh.
It's gone.
No, it wasn't you. It was your environment, not you. Sorry. I didn't mean to say you were a snake. No.
Or having a hissy fit.
It's gone. It's gone. No, this is just air or something, but it goes away in the post-production. I don't worry about it.
Okay, fine.
But it just bothers Dickson.
Didn't bother me. I was just
sorry.
alerted to it for the first time.
Okay, so then they took 4 additional koalas and did some sequencing on tissues. I guess these are available. I guess the Queensland connection there, they can get it.
Right.
And they can see in those animals new insertion sites for not only the koala retrovirus, but also the other 3. So these things are moving around in real time right now, right? So that's cool. They look at the germline in particular to see what's the situation. And these 4 new animals had 60 germline insertions that weren't present in that reference genome. 60. This thing is going crazy. Hey, a new host. Let's move around. And none of these were the same in the 4 individuals, these 4 new koalas that they're looking at. And they say these were all collected from the center of the habitat range of koalas, I guess. I don't know what that means. North to south, right?
I think so.
So did they check the offspring to see if they had identical
they
or
only
not?
had these.
That's too bad.
I figure they have some tissues stored away, right?
I mean, the recent studies.
No, they just have this. They don't have offspring.
Okay.
Just have.
Because
I'd give you a hint as to how it's.
Yeah, no, I'm sure.
yeah, and I think 2 of them they said they actually were using previously published sequence.
Right. The reference sequence or these 2
No,
of these 4?
Yes,
Yeah,
Burke
okay.
and PC were previously published.
Good thing they didn't give them names, right, Dickson?
It
Right.
is. Ernie
Be a
would be the other one I would
confidentiality
think of at least.
All right. So 60 germlines, all different. And these 4, they say, are probably at least 2 independent invasions of this virus. In other words, infections followed by expansion within the genome. So it gets in and then boom, 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 jumps all over the place. And how does it do that? So the DNA is integrated, makes an RNA copy, reverse transcription makes a DNA and then it goes somewhere else because the virus encodes an integrase, right? And all that. That's how
one of the 7 replication modes.
That's right. It is one of the 7.
It is. Mm-hmm.
So these endogenous retroviruses, the koala and the others, are all active, and they're all moving around the germline of wild koalas. It would be interesting to get more, but you just don't go out and randomly kill koalas. They're endangered, right?
Very much so.
So
very much so.
Okay, so another thing they do is compare the rates of transposition in somatic versus germline tissues, which is quite a feat in itself, but let me just give you the result. The endogenous retrotransposons, now it's kind of interchangeable term, right? Endogenous retrovirus, retrotransposon, they're moving around. They're less active in the testes, and that's where these piRNA defenses exist. So the clue is that, maybe it's being limited by piRNA in the testes. So if you believe the data that there's less transposition, less activity in the testes, then that's quite interesting. So the rest of the paper is really characterizing this small RNA response in this koala. And it's a rather complicated paper. I think there is a commentary which actually nicely summarizes a lot of it. I had to read this many times to begin.
I didn't read the commentary because that was post paywall.
But the commentary has a nice model
picture
that gets you started, and then
you can
the paper itself has a graphical abstract which you wouldn't be behind the paywall.
That actually, the graphical abstract is pretty handy.
Yeah, and then there's also another model at the end of the paper, right? Yeah,
there is, which is very complicated. I find the one in the commentary a little more accessible.
The one in the commentary is very helpful.
So now that you can take small RNAs from different tissues and sequence them, and there are signatures of piRNAs, different, how long they are. The one end is, the 3' end is methylated, 2' won't methylate, etc. But they can find they're 18 to 34 nucleotide long, which is different from si or microRNAs. So they can find these in the testes of koalas, and they look just like piRNAs that have been found in other organisms. Okay, so presumably these are reacting to the koala retrovirus and the other retroviruses. Now here's a little difficulty here. So piRNAs, in all the organisms they've been studied, they're typically made from 2 places. One they call intergenic clusters, and then isolated transposon insertions. So they're clusters of sequences that give rise to piRNAs in between genes. And then there are places where there are transposons inserted, and right there, the piRNAs are generated as well. And so they do an analysis to show basically that koalas are like other mammals. They make piRNAs from clusters and wherever transposons have been inserted. Okay. Now, what is this insertion? So the idea is that when a transposon goes into a cluster of piRNA sequences, this helps you to make antisense piRNAs. Now, if piRNA is going to attack a messenger RNA, it has to be antisense because the messenger RNA is plus-stranded, right? And this is a way of making, by integrating into these clusters, is a way of making antisense piRNAs because you can get transcription from both strands of the DNA. And so they find these in the koala genome as well. And they have this idea that these are these transposition of the transposon into these clusters helps you make antisense piRNA, which is important for propagating the inhibition, right?
So just the non-virologist in the crowd.
Yeah,
here.
I know, it's hard.
the Society of Non-Experienced Virologists, that's me. The PIs are made spontaneously. They're indigenous right? That is to say, they're not stimulated by the transposon. They're there already, but they're made in low numbers and they're made in random numbers. And then the transposon comes along and selects the one that actually matches with it. Is that what we were listening to?
It's a little more complicated. Wait to the end, okay?
Oh, okay.
I think you'll get an overview.
Okay.
I mean, we're in the weeds right now.
We are.
Wait till we get
I just want to know if it's inducible or not. That's what I'm driving at.
the piRNAs.
Yeah, that's right.
I'm sure they are.
Mm-hmm.
They're promoters driving them.
So this is almost like an antibody-antigen reaction.
Well, it's a response, I'm sure. I mean, we don't know. They don't know actually what are the triggers.
So that was my last question, but I'll ask it now. Could you
Yeah.
possibly cure an animal using CRISPR and then watch it all come back again?
Sure. I mean, you have to reinfect them, right?
Yeah, exactly.
You
They
want,
got
to
infected again.
you want to take out all the
All of
endogenous-
the endogenous, and then put the one that you're interested in back in and see what happens.
Right. So I would say you probably should do that in a fly. So you don't,
Yeah.
right? Because it's easier to do. But I also just have this feeling that there are probably fewer things that you have to take out, but I could be wrong.
But there's
about
this
that.
new editing.
Anyway, yeah, you could do that.
There's this new editing approach that I heard on the news, by the way.
Dickson. That's right.
No, I did. Did you hear that also?
Yeah, so they took
Yeah.
CRISPR with reverse transcriptase
right.
and linked them so
Simplify
that
it.
you can get single base
Right.
changes.
Correct.
And you don't have to cut twice and all this. Yeah, it's very cool. We'll do that.
You measure
In
twice,
coming
cut
weeks.
once.
You try to cut once.
Dickson, there's this amplification pathway with the Piwi RNAs
Yes.
that can help to increase their numbers that you can in some ways talk about as being inducing more
Indeed.
the ping pong amplification
Yes.
pathway.
Okay.
Yeah,
Thank you.
it's what amplifies. I didn't want to get into it because it's a little bit complicated, but
Got it.
yeah. Now they find that in their koalas, they get a lot of sense piRNAs, right? Not antisense, sense, and they want to know what's the source of this koala retrovirus sense piRNAs. Well, it's very interesting. These retroviruses, when they integrate, then the genome integrated is transcribed to make a messenger RNA. The full-length unspliced messenger RNA is exported to the cytoplasm where it then gets incorporated into virus particles. That's the viral genome. But also in the nucleus, it's spliced to give different splice products, and those give rise to viral proteins.
Good Lord.
So you splice to reach the reverse transcriptase, for example. They find that the piRNAs are derived from the unspliced transcripts. Preferentially, they do a analysis of what fraction is coming from, and very few are coming from the spliced mRNAs. They're mostly from the unspliced transcript. They say Koala retrovirus piRNAs are produced almost exclusively from unspliced proviral transcripts. So this tells you right away what's going on.
Right.
And the commentary uses the word shredding. So these full-length transcripts, unspliced, are shredded to form little piRNAs.
Wow.
And those will eventually help make antisense.
See the
So
RNA.
ice.
I deduced, and tell me if I'm wrong, I deduced that this shredding or RNA generation is happening in the cytoplasm.
I think that's part of it, yeah. Because
at the very last paragraph, they talked about, because I suddenly thought, well, if it's a way to tell spliced from unspliced, and it should be spliced, but it's in the cytoplasm and it isn't spliced, then maybe that's a signal. And that's kind of one of the possible models I think
Right.
that they allude to.
yeah, so there's something about not being spliced that triggers the shredding. And that could be in the cytosol. But they also say that this is a weird mess. It's not meant to be translated. So it may be detected as weird, and that also triggers the shredding. They talk about nonsense mediated decay pathways, which,
Right.
let's say you have a messenger RNA without a stop codon. That'll be seen right away and be destroyed by this
Right.
nonsense mediated decay pathway.
Right.
So they're thinking that somehow that's linked to the piRNA
What's
pattern.
the
So it's,
shredder?
Right,
it's
but-
essentially
but
it's
not-
a pathogen-associated molecular pattern.
Exactly.
Right.
Exactly.
But the nonsense-mediated decay is also going to be cytoplasmic
That's right. That's right.
mechanism.
Yeah. So I think this is happening in the cytoplasm. Yeah.
These are RNases.
So you want to know what's doing the shredding?
Yeah, I do.
There are these Piwi proteins that are associated,
Piwi protein.
which are like Argonaut, right? Which
They're not super small, but they're Piwi proteins.
so those are likely doing the shredding.
Right.
But they didn't show that here. That would be interesting.
Yeah,
to look at.
they speculate on that a little bit in the commentary, but this was where I really had to stop and research a little bit about Piwi RNAs, because the sense antisense bias part of it threw me for a second. Ideally, you think of these as antisense, maybe working like an siRNA or something like that, binding to the transposon and stopping it because of that complementary base pairing. And here you think, well, the sense version can't bind to its
Genome.
complement, can't bind to the genome.
Right.
What in the world is the sense version doing? And so I was confused about that for a while until I realized that the shredding and making the sense versions was the active part.
That's right.
Not
Yes.
that the sense version was going to do something.
That's exactly right. And that
Right. And then
shredding-
later, the sense version can be used as a template to make
That's right.
the anti-sense version.
Exactly. That's right. So the shredding is really the antiviral part.
So
And that
life
confused
really,
me too, but and and these spliced messages are not shredded. So there's something different, and this got people very excited. There's something different about unspliced versus spliced that's triggering this piRNA pathway to shred it. And that we don't know what that is. There are lots of possibilities which they mention.
So when they say smoke and mirrors, they actually mean smoke and mirrors now. This is like this sounds like you just made all this up.
Well, it's not. It's
No, I know. So if you were to say
you
this,
know, biology gets really amazing.
it's
the more you look.
How far back does this go? Did you say back to the Carboniferous era with insects?
How far back does what go?
The
Endogenous
system,
retroviruses
the
or
system.
Piwi?
No, the system.
The system is ancient. It's in
Ancient, you
all
say.
kinds of organisms.
Ancient.
Yeah,
So transposons were original parasites almost.
it's in yeast.
It's in yeast.
It's very ancient.
I'm not sure about that. It's a eukaryote. Come on.
Well, it's not as ancient as bacteria, but yes, but still quite old.
Yeah.
But this shredding of unspliced transcripts happens elsewhere. You can find it. They look in mouse, rat, cow, opossum, chicken, and flies. In each of them, they can see piRNAs almost exclusively made from unspliced sense strand transcripts.
Wow.
Right.
Wow.
So it's old. It's deeply conserved, is what they say. Deeply
Deeply.
conserved.
That's right. As opposed to meh.
And they also look at a mouse endogenous retrovirus, a different virus of mice, and same thing. Preferential processing of unspliced proviral transcripts to give these piRNAs. And so this is the contribution of the koala. They see that this bias, and they attribute it to the shredding of the full-length unspliced transcripts. And now it makes everything else make sense.
This is the DNA police.
It is the DNA police.
Yes.
I like it.
This is all
a good way to put it.
but it's all at an RNA level, though. You know.
Yes,
Did you
but
say RNA
it's-
police?
No,
RNA
no.
You
Dickson.
could, but it's policing what happens to the genome.
Policing
The
the
DNA has
Yeah,
to make
it's
this
gotta-
protein. It has to make the PI protein that does this.
Oh, yeah, that's true.
So therefore, it makes its own police force that goes out and keeps the transposon limits down.
Yes.
Not off, but
So
down.
the name of this commentary is piRNAs Rise to Rescue Koalas. And I thought for a minute, well, they're not really doing a good job because the koalas are getting infected, but they're protecting the germline.
Right.
And so the koalas are still reproducing, because without this, they would probably be out of existence.
And I would also add that PIs get shredded routinely by NIH grant reviews.
Yes.
Yes. And
Very
Dickson,
good.
I've been thinking a lot in my lab about DNA sensing and things like this. So do you mind if I call my lab the DNA police?
Would you please? Would you please?
Yeah, you can make buttons. You should make buttons.
Please.
I think I will.
Make a button and give everyone who joins the button.
I'm good with that.
My friend Mark Martin, he teaches undergraduate microbiology at Puget Sound, and he says every class he teaches, he starts out by giving everyone buttons.
Fantastic.
And he said they love it, they get motivated, and they're designed by the class, right? So,
neat.
And he's given me a bunch of them. They're very cool. DNA police. Well,
Police.
you know the.
I'm going to take it and run.
You know the Sting police, right? And that's funny.
Yes.
So when I was in Denmark, I was talking to one of my guests about DNA sensing, and he said this one protein is called Sting, and he made some connection between the person who discovered it and the police.
Right.
He said,
Wait a minute.
It's from the UK, so maybe that's why he called it Sting.
Right.
But no, it's not. There's an abbreviation, as you know.
Yeah,
Yes,
like
it's
an
the
Englishman in New
stimulator
York.
interferon
Yeah.
genes.
Yeah, I like that. That whole discussion about DNA sensing in Denmark was good.
He was a funny guy. He had a sense of humor.
Sting would like that episode, by the way.
That he's named.
That he's associated.
Well, a
with,
protein is named, but it's not. It's got its own thing.
No.
So flies and yeast do the same thing. Preferential processing of unspliced transcripts, and this now is a mammal which is doing the same thing, and koalas and the mice and so forth.
Could also have a message in the bottle. In this case, it's a message in a test tube. Sorry.
Message in a bottle. Fruit flies you grow in bottles.
You know that.
So that their proposal is that this whole PI response has innate and adaptive phases.
Got it.
So when a genome is first invaded by a retrovirus, the proviral insertions, they're transcribed. The inserted retroviruses, you get spliced and unspliced transcripts. Something about the unspliced transcript triggers the PI pathway so that they start shredding.
So that's the innate branch. The PAMP sets off this response. It shreds the RNA.
How long-lived are these small PIs? That is what is the turnover rate?
I'm 65, 66.
No. Okay. No,
And I'm pretty sure.
I'm sorry I actually started that line of reasoning, but so, you just can't keep on making these things, right? I mean, can you?
Sure, they're regulated. No, you don't keep making it.
No,
all
nothing.
things, right?
Well,
everything's regulated to a certain extent.
To a certain extent, yes.
So I'm sure these are induced. And so they say, they talk about what might be the trigger for production of this. And, it could be a stress response. When foreign DNA inserts into the genome, it triggers stress response pathways.
So the spliceosome has a unique anatomical location within the cytoplasm of the cell, yes?
Well, the spliceosome's in the nucleus.
Within the nucleus.
Yeah, you said cytoplasm.
Did I say so? I'm sorry. I didn't mean that. But
okay.
But how do you prevent splicing if the shredding occurs in the cytoplasm?
You don't prevent splicing.
Yeah, some of the message gets spliced.
So some of it gets spliced, some of it gets sent out.
But some of it's not spliced because the virus eats the packaging.
Yeah, that's the viral genome.
So that's a viral choice, if I may.
Okay.
Some of it, splicing can be regulated, and there's all kinds of signals that do that. A fraction of the transcript is unspliced, but normally that would not get out, okay?
So virtually all of the host cell RNA gets spliced.
Yeah, and that's a signal for
foreign RNA.
That's a signal for nuclear export, actually. So it's very rare for an unspliced message.
So
it
may be what's setting off the shredder in the cytoplasm.
Yeah.
So that's the innate part.
Right.
And they say
the adaptive
these little plus sense they say primes the adaptive response, which is triggered by proviral insertion into a cluster or mini clusters, and I'm not quite sure what happens. So proviral insertion is what is this cluster? What's in this cluster?
So the cluster, I think, is where other piRNAs are encoded, right?
Right.
And so when by my interpretation of this was by chance at some point, if this thing's jumping around the genome, it's eventually going to jump into a cluster.
Okay, and then you'll get transcription from both strands and make piRNA.
Right, that cluster gets processed specially. Transcribes both strands to make the antisense.
Yeah, and that can then...
The antisense is then the adaptive form, which is going to actually silence the the retrovirus.
And you have memory because it's integrated. The sequence is there forever to protect.
So that is then in the germline, and that protects future generations against the virus.
And that protects against jumping or new invasions, which we see are happening in the koala genome.
Right. So eventually, one hopes the koala genome will have this jump into the right spot, and then it'll turn on their adaptive immunity, and then they will be immune from this. It'll just be like their other endogenous retroviruses.
Yeah. And they make the point that during the shredding phase, you don't inhibit the transcription of the provirus.
Right.
And so it can still move around, and eventually it will move, as you say, into a cluster, and then that is the end.
Right.
Then you make antisense, and you can really silence these
right. So right now it's
proviruses.
tamping it down by shredding its genomes in the cytoplasm,
and you can't make viruses. Then
you can't make viruses from that, but I guess enough is still getting through that this thing is still going to spread too, right?
Well, you're always getting transcription of the full length, right, and that's going to get out. It won't make virus particles because it's shred, but eventually it could still transpose. It can still transpose.
Isn't the virus still spreading horizontally?
Not from the germline. It can spread from somatic tissues.
Right. Okay. Right.
But you want to avoid germline transposition, and this is this shreds, so you can't transpose. You can't make viruses in the germline, and eventually you get a permanent record of this sequence.
Because the Piwi system is in the germline.
Exactly.
So there are no unspliced host RNA transcripts.
There probably are, but they're few. Right?
And here's where I'd like to jump back in with there are two possible models of what's going on here, where this pathway might be functioning as an alternate cytoplasmic quality control system.
Mm-hmm.
And so the support of that is the fact that there's this UPF1 that's part of nonsense-mediated decay, has some similarity. But in other words, there could be some host cell messages that get out and are unspliced, but this as a quality control mechanism that would take care of them.
But wouldn't you find those little fragments also then?
Yeah, you would if you looked for them.
They should do that.
And the available data cannot distinguish between these alternatives, but the models are testable and provide a framework for further analysis.
Indeed. So I was thinking that if all of the host RNAs were spliced within the nucleus, and most of the transposon RNAs are not, then there's a gatekeeper that keeps all of the host RNAs within the nucleus before they're spliced.
Mm-hmm.
So this virus has worked out a way of avoiding that gatekeeper by getting its unspliced RNA out. So what is that mechanism?
Well, it depends on the virus.
There
are retroviruses that do it. There are herpes viruses that have to get out unspliced. But for the retroviruses, I don't know about these gamma, but I know HIV has a small protein called REV that binds to the genomic RNA, which is not spliced, and allows it to use the nuclear export pathway to get out. Because normally, when you're spliced, the proteins that remain on the spliced mRNA are recognized by the nuclear export system, and that's how it gets out. If you're not spliced, you don't get exported unless you have a special signal like these retroviruses or other viral.
So those are the
RNAs.
deputies.
Yes.
Right? Anyway, Vincent's textbook has a really nice figure that shows those two that I use in my class.
Okay.
So you have to take a look at that.
Kathy, it's interesting. They call this failed splicing. But I think that's a cell-centric view.
Right.
Because for the virus, it's not failed splicing.
No,
it's not failed. It's needed to get genomes out. And for herpes,
successful genome making.
So it's interesting.
So it avoids being coupled to this escort protein.
If you don't splice, you're not recognized by the nuclear export machinery.
And what is the molecular basis for that recognition?
Lots of little proteins. Nups on the membrane.
I mean, what is the... No, I don't mean that. I mean, isn't there a sequence that is always recognized by the protein?
Well, there is a nuclear export sequence, but then there are a variety of proteins that recognize it, and also the splicing proteins that are left on the spliced RNA. And
how many escort protein types?
Well, don't say escort, because that's a different pathway in the cytosol.
No, I mean just to get out of the nucleus.
How many?
Yeah, they're different. How many different ones?
Well, I see this picture in my mind. There's at least a dozen on that picture that I'm looking at.
So the nuclear pore has to have a recognition signal for each of those?
No, just one or two. Yeah, it doesn't need to recognize all of them. One gets it to the pore and attaches to it and gets shuttled through. And of course, it's an energy dependent mechanism. You have to have a gradient.
All right.
And it's a very interesting mechanism that's involved. Yes.
I'm creating a cartoon in my mind.
I think that's interesting. They call it failed splicing. So then they go through some similar results in flies, unspliced transcripts producing piRNAs, and they say piRNA biogenesis in the fly germline and unspliced proviral transcript export in mouse somatic cells thus co-opt the same host factors to bypass nuclear quality control mechanisms and export unspliced transcripts, suggesting a common origin. So this piRNA and splicing that they're suggesting have a common origin. It's pretty neat.
Wow.
Yeah, so that's what's cool about it, because you have this neat trigger, which is an unspliced transcript that gets the whole thing going.
I want to ask Brianne a question, because they make this really strong analogy to what I consider sort of classical immunology in all their terminology. Mm-hmm. Okay, so they talk about the bypass splicing generating a molecular pattern that's recognized in an innate genome immune response. And then there's, later they use the word that this initial sense strand piRNA production primes the adaptive genome immune response. Right. So they talk about the sequence-specific adaptive immunity generating a memory of the genome invader. Do you think this is going to confuse people to use this terminology? I mean, I think if you understand that we're talking all about just genome stuff here, it works really well. But I just was thinking this could confuse undergraduates.
I think it possibly could. When I really thought about their use of innate versus adaptive, it helped me understand the model.
Mm-hmm. I found it helpful too.
Because the idea is that with innate, it's just any transcript that's not spliced.
Right.
Any transcript that has that pattern is getting degraded. There's nothing specific about it. And in the adaptive, you have this nice antisense that is going to only silence its complement. Mm-hmm. And so it gets at the specificity versus non-specificity aspect. I can certainly see some ways that it might be confusing, but given the number of things where I've read about things like restriction enzymes are bacterial innate defense and CRISPR is bacterial adaptive defense, it seems like this sort of idea of genome protection and these terms has been catching on.
Okay.
The commentary calls the innate phase cis silencing.
Right.
And the adaptive phase trans silencing.
I think that's more confusing.
I do too.
Confusing. Much.
Okay. But it's working on the genome versus
No, I think the innate versus adaptive terminology makes a lot more sense, and it's conceptually consistent. It's not like lifting a phrase and misusing it, like a quantum leap or something like that. This is a totally appropriate use of those terms from classical immunology, I think.
The other thing that's worth considering here, so this is basically an antiviral defense for the germline based on small RNAs. And if you've listened to TWiV for a while, you know that there has been a controversy on whether small RNA antiviral defense exists in mammals. It certainly does in plants and invertebrates, no question. But in mammals, it's not clear. Ben Tenhoover, when he was last on, talked about that. There are a few papers, however, which say it does exist in germ cells, only in germ cells in mammals. So maybe this is what that is. Because here the data are quite clear that there's something in the germline, sort of like an RNAi pathway, piRNAs, right, for antiviral defense. I think that's quite clear. They show another example in a mouse and a mouse retrovirus. So maybe this is, they say, does this first line of defense protect invaded germ cells long enough for the infected animals to reproduce before they succumb? Does it buy time for the individual, but not for the individual, but for the species? So that's interesting to think about because, depending, most people you talk to say, there's no antiviral RNAi in mammals, but maybe in the germline there is.
Yeah, I thought those same kind of things when I was reading this.
Their last sentence, these data uncover an exciting aspect of piRNA biology and shed light on the risks of acquiring a novel genomic resident that changes the identity of a species forever. That's pretty dramatic.
Yeah.
It could kill it, too, right?
Yeah.
So that's why it's interesting, because you can do it in koalas. It's happening real time, and you can see how it's
koalas have
defended.
a very low reproductive rate, as a lot of marsupials do. But this
so it means
particular,
that
it would have to.
They're not fecund.
They're not fecund, and I mean they're getting hit in every direction at once on this, but they're older when they reproduce, or they live long. Let's put it this way: they do live long, and
do they prosper?
They used to. I mean, look at how long it took them to get from the north to the south, for instance. If you look at their rate of migration, it's very, very slow. They're very docile.
They don't move around a lot.
They're like a sloth that's not like a sloth, basically.
Yeah, they're a little more mobile than sloths, but they'll spend, a koala will spend its whole life in one or two trees.
Exactly. So
just that's where it is all the time.
To show you an example of how vulnerable they are to their own behaviors, a lot of koalas die by falling asleep in a tree and falling out.
Oh, that's sad.
It is.
I hope someone's taking the ones that are uninfected and bringing them elsewhere.
I don't know that.
Like
that's possible. Like they were for the
many zoos have koalas, right?
For the Tasmanian devils are doing that too.
Speaking of slow, have you ever seen a slow loris?
I have, actually, in the Bronx Zoo. There used to be one there.
Very slow. Okay,
it's true.
Let's do some email. I think, Brianne, you should take that first one.
Okay. Diane writes, "Hi, Professors TWiV. So for the first time ever, I got the flu. It has frankly sucked, and I hope I never get it again." I agree, Diane. I got it 4 days after my annual flu shot. Now, I'm a devoted nurse and listener to TWiV, so I know that the flu shot didn't give me the flu. But one of my good buddies, a pharmacist to boot, has decided that it was the shot that gave me the flu, and furthermore, he's not going to get vaccinated against flu this year. I'm so irritated I could spit, but that will probably trigger another gigantic coughing fit. I also agree, Diane. Yeah. I'm going to nag him. I'm sure that will do great. #sarcasm. So I have a couple of highly speculative questions, if you'd be so kind as to indulge me. First, what are the odds that a flu vaccine would be released with live virus? In the shot, of course, not the live attenuated intranasal vaccine. Second, if in the presumably highly unlikely scenario that the shot contained infectious virus, is there any possible way the virus can make its way over to the airway tissues from the deltoid and start infecting? I don't know viruses well enough to know, but it seems really unlikely. And of course, with basic stats, we know that the possibility of 2 unlikely events is markedly higher than a single unlikely event. I hope these questions aren't too dumb. Please feel free to ignore this if it's just too silly. As always, thank you for a terrific podcast that I never miss. I appreciate all the time you put into it, your camaraderie, the groan-worthy puns, and as always, the science. Diane. From Seattle, where the weather is happening, but I have not been outside in days because of the dumb flu.
I think she means the possibility of 2 unlikely events is markedly lower than a single unlikely event.
Yeah.
Yes.
Lower, yes.
Because it's multiplied.
So those are good points you make. So what are the odds that it would be released with live virus? I think very low because every batch is tested for infectivity. And
extremely low.
And this happened once in vaccine history. That was the first polio vaccine released in 1955. Some batches were not completely inactivated because the kinetics of inactivation were not interpreted properly. We now know this, and every batch is tested. But let's say
because of that event, the vaccine industry is frickin' paranoid about this.
Mm-hmm.
Like this is the failure mode that everybody is keyed up about in manufacturing a killed vaccine. Is it killed? And that's tested thoroughly.
Non-infectious, not killed. You don't kill a virus that's not
so
alive.
let's say on the off chance this is an intramuscular injection.
Right.
You got some influenza virus infectious in your muscle. It will not replicate there because the proteases needed to make infectious virus particles are not present anywhere except in the lung. So it's gone. It's not getting anywhere. And if a few particles somehow got into your blood and somehow made it to the basolateral membrane in the lung, which is all really unlikely, it would still have to cross into the basal. So I think too many unlikely events. Intramuscular inoculation will not give you influenza. It just cannot happen.
I agree. And sometimes I hear people say, well, I always get the flu from the flu vaccine. And I wonder, really,
wait a minute.
All of these multiple unlikely events happen to you and only you multiple times.
Why do you keep going back if that's the case?
Yeah. It seems difficult to imagine how
I don't believe it.
This happens frequently to you, but not to anyone else to a point that people would notice.
Mm.
Maybe they like the flu and they go for their vaccine just so they can get it.
Right. Now, what is likely happening is that they get the shot, their arm is sore, they get a little bit of an immunological reaction to the shot that feels like they're starting to get sick, and they say, oh my gosh, I got the flu from the shot again.
Yeah, exactly.
That's it.
But no, you didn't get the flu, and it's, yeah.
I'm looking for a tweet that I liked, but I can't find it, that I thought she might be able to use some phrase like that on the pharmacist. It's basically something like, by not getting the flu shot, could you have been in the chain of transmission that gave my child the flu that killed my child or something? I mean, it was pretty dire, but it was much better phrased than that. And if this is a pharmacist who actually sees people, that are rotating through the pharmacy wherever he is, he could be giving it to, really highly susceptible people. And it's just not, I can't think of the word.
Ethical.
Yeah.
It's irresponsible.
Like it's irresponsible. It's just not responsible. Yeah. So, and also the fact that, it's highly unlikely that the flu shot could give you the flu. So, yeah.
The next one is from David, who sends us a ProMed mail post about a child in Oregon who contracted tetanus.
Yes.
6-year-old boy who had received no immunizations. He got a forehead laceration and 6 days later developed jaw clenching, muscle spasms. Got a diagnosis of tetanus, got care and was rehabilitated. It was told that his parents should immunize him, but they refused. Routine administration of a 5-dose DTaP series. DTaP is recommended for all children. Right. So there's a footnote to this report. It says, unlike most diseases, in tetanus, disease does not result in tetanus immunity. Clinicians should begin or continue active immunization with a tetanus toxoid-containing vaccine as soon as the person's condition has stabilized. Unfortunately, this was refused by the child's family.
An important, I think this is an interesting point here too. The boy required 57 days of inpatient acute care, including 47 days in the ICU. The inpatient charges totaled $811,929, excluding air transportation, inpatient rehabilitation, and ambulatory follow-up costs.
Amazing.
Yep. So this is coming out of your insurance premiums.
And the tetanus vaccine would be $50, right?
Instead of being directly billed to the right. idiots who were responsible for it, who were his parents, who refused to have him vaccinated.
$50 or $800,000.
Yeah.
And there's a point later in the ProMed that says that Adult tetanus cases included hospital charges from $22,000 to over $1 million.
Yeah.
Yes, that's amazing. That is amazing. And I'm sure the kid didn't like.
And not every. I mean, okay,
No.
tetanus shot you're supposed to get every 10 years, but not everybody does. But if you get a serious wound, you get it checked and get a tetanus shot. That's when you get it, because you can actually prevent this from happening if you get a prompt vaccination.
It's like rabies in that sense.
Yeah.
But not in the other sense.
No.
We now have a letter in the continuing series about quasi-species.
Oh, just before you do that, I found the tweet. I want to read it. Sorry.
Okay.
Jude died from the flu on May 6th, 2016. Everyone who gets the flu gets it from someone else. How many people had it before it came to our home in May? What if just one of them had taken more care to prevent contracting and spreading the flu? What if?
Yeah, it's great.
Yeah.
It does kill people.
Mm-hmm.
Alan, can you take Lucian's?
Sure. Lucian writes, thank you very much for the discussion on quasi-species on TWiV 567. However, there was some grumbling about selection acting on groups when selection should only act on individuals. My understanding was that individual selection was still occurring. In classical population genetics, if one sequence is more fit than another, it will eventually become fixed in the population. However, this assumes that a sequence can be faithfully reproduced. It's an assumption we often don't even consider since it's often so true, but in RNA viruses, sequences cannot be faithfully reproduced. Take an idealized example. Let's say there's a specific sequence, sequence A, that is the most fit. The naive assumption is that this sequence should become fixed as the virus replicates. But none of this uber sequence's progeny will have that same sequence. RNA polymerases make too many errors. We can call the collection of sequences that resemble sequence A quasi-species A. I like that. Consider another sequence, sequence B. It's not as fit as sequence A. We can likewise call the collection of sequences that resemble sequence B quasi-species B. If the average fitness of quasi-species B is higher than the average fitness of quasi-species A, we should expect quasi-species B to outcompete quasi-species A. It does not matter that one of the sequences in the A collection is more fit than any other. It cannot spread without high-fidelity polymerase. In other words, if sequence A's fitness is severely compromised by any mutations, but sequence B's fitness is more tolerant of mutations, we should expect the collection of sequences that resemble sequence B to outcompete. Group selection is not occurring, but the emergent behavior appears like selection is operating on populations, not individuals. This was my understanding of the concept. Yet another, I suppose, to throw into the ring. I think that's a really interesting point.
I agree.
That's a good description.
Yeah, I think that's really well put.
All right. Kathy, can you take Benedict's?
Sure. Benedict writes, Dear TWiV Consortium, concerning the uncertainty y'all expressed when deciding whether the Berlin-London patient's HIV could have been cured since everybody develops X4 tropic virus. That's not necessarily correct. I would assume that both patients were tested for virus tropism, and the results showed that the overwhelming majority of the virus strains were R5 tropic. HIV strains often, but not always, tend to develop X4 tropism late in the course of the disease. That is, once the patient's CD4 cell count lowers. Exceptions to this rule include fresh infections with X4 tropic virus through IV drug use, thus eliminating the bottleneck which exists when infected through mucosal surfaces, or patients dying with only R5 tropic virus strains. No exact rules are known as to why or when X4 tropism develops, but it is not given that it has to develop. It is not a given that it has to develop during the course of the disease. Some virus strains are even dual tropic, meaning they're able to utilize both co-receptors for entry. While no one knows why it arises and exact mechanisms are also not defined, the electrostatic charge of the V3 loop, a part of the fusogenic GP120, as determined by its amino acid composition, seems to play a considerable role. In silico tropism assays almost exclusively make use of the V3 loop sequence. I hope to have been of some assistance. Yours truly, Ben. P.S. I've been backing y'all since you introduced your Patreon account, and I've yet to run into serious financial issues. It's cheap, people. Just do it. P.P.S. The weather discussion is great. Leave as is, please. It's already dark here in Austria, but today was a gorgeous autumn day, sunny with just some white wisps in the sky, 19C tops. All the leaves are turning now, just beautiful to behold. PPPS, be nice to Dickson. I know you've made an effort the last shows, just a gentle reminder.
I'm sitting here with a smug look on my face.
Yeah.
Okay, so the point here is that sometimes you can have R5 virus, and therefore if you get a bone marrow transplant with a CCR5 negative donor, then that will get rid of your infection.
Right.
Mm-hmm. And we have another response to the 4-5 issue from Alexi. Dear TWiV team, a question from TWiV 569 on possible development and use of a CXCR4-based method of HIV infection cure that would be similar to those used for Berlin and London patients is indeed interesting. However, I would like to emphasize that methods of cure or achieving long-term remission used for these two patients are exceptional and are not seriously considered as a strategy for essentially any HIV patients. And yes, I had meant to say that, and I woke up one night saying, I didn't say that. Right. That's the most important thing, that a bone marrow transplant is not going to become routine. No. So thank you very much.
No.
Both Berlin and London patients had life-threatening forms of cancer, and that was the reason why an irradiation or chemotherapy followed by a transplant of donor hematopoietic stem cells was used. If these patients did not have cancer, they would never have been treated that way. Such a treatment is extremely risky, expensive, and it is very difficult to find an optimal donor. So even if a few more...
And pretty unpleasant, too, I gather.
Yeah.
Even if a few more of such cases ever appear in the future, they will remain exceptional proof of concept cases. However, active research is ongoing on possible strategies to delete or mutate 5-4 for scenarios that would not involve total destruction of original myeloid stem cells by irradiation, and that would use autologous instead of allogeneic stem cells for transplantation, linked to a recent review. For both patients, the reason for choosing a CCR5 delta 32 version of a CCR5 gene to substitute for wild type was that this is a naturally occurring mutation known to protect, in its homozygous state, about 1% of Caucasian population from being infected by R5 tropic HIV strains. 16% mutation frequency mentioned by Vincent includes heterozygotes, while only homozygotes are fully protected. However, no such naturally occurring protective mutations are known for CXCR4. Moreover, deleting or mutating CXCR4 is lethal in mice. Even if such a mutation of 4 is found that protects from X4 tropic HIV strains and that is safe for a patient, it should never be used on its own, either in a clinical context similar to Berlin-London cases or in a context unrelated to cancer. The reason is that in the majority of cases, R5 tropic or R5X4 double tropic strains, but not X4 tropic strains, are transmitted during the initial infection, thus establishing themselves as transmitted founder strains that give rise to the infection at the organism level. For example, Wilkin et al. Found that in ART-experienced individuals, 50% viruses were R5, 46 R5X4, and only 4% X4. The prevalence of X4 strains seems even less in ART, antiretroviral therapy naive individuals. Therefore, the use of any possible CXCR4 mutation alone would be completely irrational and even unethical, and only a combination of mutations of both CCR5 and CXCR4 should be used, provided that such a combination would be safe and efficient. So while this question is indeed very good for an exam, I would say students would be able to give the best answer if they are already familiar with the concept of a transmitted founder virus and with the prevalence numbers for R5X4 and R5X4 strains. Finally, about the question of what was the HIV tropism in Berlin and London patients. For Berlin patient, an original sequence analysis by Uckel et al. Predicted that this patient had pre-existing X4 strains. However, Simons et al. Performed phenotypic tests on co-receptor usage and concluded that those strains were CCR5 dependent. Links for both. This is a good illustration of the importance of doing phenotypic assays and limitations to using viral DNA protein sequences for co-receptor usage. I don't know why the authors of the review that I linked to above, Allen et al., don't cite Simons et al., writing instead that the Berlin patient had X4 strains. Maybe they missed the Simons study. For the London patient, Gupta et al. Established the absence of pre-existing X4 strains. Another link. Even though the exact mechanism of virus clearance remains unknown, for both cases, Gupta et al. Suggest mild graft versus host disease as an unconfirmed hypothesis. Hoping to keep it short. I did my best. Keep up your great work. Best regards, Alexi. That was great.
Yeah.
Very nice.
Very good. Alexi, can you come tell my students about all of that?
Sure. He probably could if he's here in New Jersey.
It's true.
Okay, time for some picks. What do you got, Brianne?
So I recently learned about this book by David Goodsell, who I've recently started following on Twitter. It's called The Machinery of Life. It's a small book that goes through sort of some basic molecular and cell biology, but he has illustrated or done computer-generated graphics to demonstrate the things that he's describing. And the artwork in this book is just beautiful. And I think that it provides some images that we don't often see, because all of these images show things like the human cell with compartments, which I'm opened up to right now. And you see this great density of all sorts of proteins in the cytoplasm, which we don't usually see. We often see the cytoplasm pretty empty. And so I think that this gives a really nice understanding of what we might see in the cell in real life. I've had a few students flip through it as they've seen it on my desk, and they all felt like they really understood quite a few things in how cells work much better by looking at these images. There's a great chapter on viruses that shows the polio replication cycle at 1,000,000x. And influenza budding. And I've just really enjoyed this book quite a bit since I've had it.
I did the Amazon look inside, and they have a cool picture of the GAPDH enzyme from E. coli, spinach, and human, and it shows you how similar they are. It's really pretty.
Yeah, all of the images are great.
Someone stole my copy. I was trying to find it for you, Dickson, but it's gone.
I'll just have to imagine.
But yeah, it's a good one. If you look in our virology textbook, we did borrow a Goodsell image to show the crowded cytoplasm.
Crowded.
It's very crowded.
At the molecular level, though, it's like
a Japanese subway.
No, I don't think
hour.
that's true. I think that if you were the size of that molecule, you'd say, where the hell are all the rest of the stuff around me? Like you look at airplane traffic and you think that everybody's going to crash into each other. But in real space, it's a lot of room between things.
If you go to the protein data bank, where they store all the coordinates of protein and virus structures, they have a molecule of the month, and he does all the artwork for that.
Oh, nice.
He's got a distinct style. In fact, in the program I use to display structures of proteins, there is a selection for the Goodsell style.
The Goodsell style.
Because he's got a very specific way of outlining atoms and so forth. Yeah. It's very cool.
Yeah, well, I think it's really useful. It's a great book.
He has a terrific last name for that also.
Goodsell.
Yes.
Yeah, it's a good sell. Alan, what do you have?
I have another art item. This is the U.S. Department of Agriculture's Pomological Watercolor Collection. So if you go to this thing, the page I've linked to is their front page. You just click on the content, 7,584 watercolor paintings, and you can search this archive that they have. These are paintings that were mostly made in the early 1900s. When the USDA was trying to promote people growing appropriate crops, and especially fruit crops for for their land type all across the country. And at the time, reproducing color photographs was pretty much impossible. So they had artists paint watercolors of these various fruits and fruit trees and fruit plants of all kinds. And it's just this amazing collection of pretty much any kind of fruit that you could grow. Everything from pears to cashew nuts, and how it looks, and just really a great image resource. And you can download, since it's USDA, it's all public domain. Use it however you like. Download high resolution versions, and some of them are just really beautiful.
They are pretty. Yeah.
Yeah.
Nice.
Dickson, what do you have?
It's not the interface to the site is not the most gorgeous, but it just search up what you want.
Right. This is an award for the wildlife photographer of the year, but it's it's an English award, and it's given out by the Museum of Natural History. And it is, you can see a lot of this and you get kind of blasé, that's a great photograph. The first winning photograph of this was just stunning. I think he took it from a drone, it looks like. I don't know how you would get that image otherwise. It's a fabulous picture of migrating antelope. They're a very unusual strain of antelope that lives in Mongolia and the steppe region of Asia. And they're migrating across a lightly snowed series of undulating sand dunes, basically. And one of the sand dunes has got an open space to it. I don't know how he got that to work out. It's just amazing.
It's an amazing composition.
I mean, he had to be there at the exact moment that this thing happened. And to be there in the moment is the whole secret to becoming one of these great photographers.
That's right. You have to be there at the right. You have to be very patient.
You've got to. And take lots of pictures.
And it says in the little mini bio, he's been living and working on the Qinghai Tibet Plateau for more than 40 years.
That's right.
And so
exactly right.
And he just goes there all the time photographs these guys.
He has another one in there that got an honorable mention.
Okay.
So he had 2 photographs selected. It was very unusual, basically.
These are great.
Very
They're
cool. Beautiful.
And Kathy, I'm sorry, let's do this one together.
Right. Well,
we did it together.
Well, I have to tell you that my colleague Akira sent this to me, and Akiko, his wife, had found it, and Akira sent this to me, and I said, coincidentally, I just saw last year's exhibit that came from the Natural History Museum in London to the Field Museum. So last month when I was in Chicago,
nice.
I saw the 2018 exhibit live and in person, and it was phenomenal. And so this was just great to see this one. I don't know if it'll come to the Field Museum again or if it'll come somewhere else in the United States, but they are an amazing set of photographs every year.
Yes. And the level of expectation has gone up so high. Why? Because you can take all the photographs you want, images you want with a digital camera, but you have to live this life. I mean, these people put their lives in danger often to get the right photograph. Or the right image, I should say.
Put my life in danger every day driving.
Yes,
here.
I know you do. You should start taking pictures.
Yeah, maybe I should make it worthwhile.
Driver of the year. Given posthumously, of course.
the WAPO picture is amazing.
Yeah. Yeah. That one of that's right.
Oh, too bad it ended up badly for...
Well, everybody got to eat.
One of them. Everyone has to eat.
Everybody has to eat. That's
but
right.
you could become a vegetarian.
You could.
Vegan. Cool. Kathy, what do you have?
So as an alternative, I picked 2 websites, Diversify Microbiology and Diversify Immunology. And these were started by 2 postdocs in our department, modeled after at least one of them that I can remember, which is Diversify EEB, Ecology and Evolutionary Biology. And I think the other one might be Diversify Chemistry. And so these are sites where you have to self-nominate. You can't nominate somebody else. But the Diversify Microbiology site aims to identify PhDs in microbiology who are women and or from a group traditionally underrepresented or underserved in the sciences. And they give examples there. And the idea is that studies show that the human brain has a strong ability to recognize and react to information, but a fairly poor ability to recall information from memory. And so if you're working on setting up a meeting or doing some kind of search or something like that, if you could look through a list, that would help stimulate you and remind you. So what happens when you click on the Diversify Microbiology public list, you can go, you can open it in a new tab, and then it opens as a Google Doc, and you can sort it by yourself temporarily. It doesn't sort the pre-existing document. And the only disadvantage is that they set this up not by first name, last name. So if you sort, you're going to be sorting by first names. But you could sort, for instance, by field. And then you could identify all the virologists or all the mycologists who have self-identified in this list. So there's one for microbiology and there's one for immunology.
Hey, look, Brianne Barker. Who's that?
Yay.
Cool. I don't know. I've never heard of her.
The immunology list needs to get a little longer here. Not very long, right?
Yeah.
Mention it on Immune.
We will.
This is good. So you can sort by a career stage. If you're looking for people to chair a panel or something, you can look for more senior people. If you're looking for somebody more junior, you can scroll down. This is great.
Nice. Very good. Yes, I will pick it on immune. I can do that. I'll cross pick.
Yes.
It's a new thing on podcast, cross picking.
All right.
That's cool. My pick is a book by Christopher Sexton called Burnett, A Life. It's about Sir Macfarlane Burnett.
Yep.
He won a Nobel Prize for medicine and immunology, but he also did a lot of virology. In fact, reading Erling Norby's book, they couldn't figure out what to give him a prize in. He did so much. He was clearly the most celebrated Australian scientist of all time, and this is the story of his life. He was a scary guy. He was very strict and serious. But he did good work. And I think in the end, he did support people nicely. So it's a cool book if you want to learn about him. We have a listener pick from Justin. He sends an article from Science Alert. A worm with 3 sexes has been discovered thriving in a nearly lifeless lake. The famous Mono Lake in California, full of arsenic, from whence people once thought they had identified life with arsenic instead of phosphorus, which was all wrong, of course.
Exactly.
But now they have a nematode with 3 sexes. How would that work, Dickson?
You're asking the wrong person.
You don't work on nematodes?
I do. But all the ones I worked on had 2 sexes.
So what happens here, they do explain it. Let me see if I can find where they explain this. Okay, so this is, they discovered 8 species of worm in the lake. One of them is weird. It can survive in a dose of arsenic 500 times what is humanly possible. Mm-hmm. It has worms of the female sex. And the arrangement...
Most other nematodes only have hermaphrodites and males.
And it also has weird
that's not true,
arrangement of genital papillae.
Sorry, I don't disagree, but the ones I'm familiar with, let's put it this way, the parasitic nematodes have clearly cut male and female, except for strongyloides. Strongyloides is a weird one.
This one has male and female.
I think they're referring to free living.
Oh, absolutely. Right. Yeah, absolutely.
This has male and female, and it also gives
right.
birth to live offspring.
Right.
Which is typical. Okay.
So as hard to understand as that is, on the news earlier this week, I heard about the Paris Zoo is showing people what they are calling a fascinating blob as an organism.
Yes.
It's Physarium.
That's right.
Polycephalum, and it has 720 sexes.
Exactly. All at once.
Okay. All right. TWiV 571 microbe.tv/TWiV for all the show notes. TWiV at microbe.tv if you would like to send us questions or comments. Microbe.tv/contribute to give us a little bit of money every month via PayPal or Patreon, as little as $1 a month to support us. We'd appreciate it. Dickson Despommier can be found at thelivingriver.org, parasiteswithoutborders.com, and many other properties. Thank you, Dickson.
You're welcome.
Who fished this week. He caught a lovely cutthroat. No,
brook trout.
Brook trout.
I'll send it to the gang. I'll put it up if you'd like. Mm-hmm. So it's the state fish for New Jersey, although it's a kind of a joke because New Jersey extincted the native strain of that trout many, many years ago, and now they just stock it. But this one was caught in New York, and it's probably a live birth in the stream kind of fish.
Many sexes of that fish are there?
No, but they do change according to the temperature of the water, sometimes with the magnesium contents. And fish sexes have a way of changing. The barramundi, by the way, I don't know if you've read much about that. It's a good eating fish from Australia. It starts out as a male, I think, and then goes out into the ocean and changes to females and then comes back and spawns.
What is that called?
That's, to do both.
Maturation?
No. If you're born in fresh water and
changing your sex, what is that called? It's not anandromous. But there should be. No. I think anandromous.
Anadromous.
Anadromous. Okay.
No. It's. I don't know if they have a word for that or not.
Okay.
A protandrous hermaphrodite.
Protanderous?
Protanderous.
I beg your pardon.
The sequential hermaphrodisian. You're first a male, and then you change into a female.
Brianne Barker's at Drew University on Twitter. It's BioProf Barker. Thank you, Brianne.
Mm-hmm.
Thank you. This was a lot of fun. Hopefully I'll even be able to breathe next time.
Recover. Wasn't the flu shot, though.
It was definitely not the flu shot.
No.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Alan Dove is on Twitter as Alan Dove. He's also at turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASV and ASM for their support, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.